Mutation is a sudden change in the DNA sequence. The effects of mutations can vary widely from being beneficial to having no effect to having lethal consequences, and every possibility in between. DNA mutations are changes in your genetic code. DNA is genetic material used to code for certain physical characteristics. It's made from 4 different molecules called bases. The complete genetic code for a human contains billions of bases. When those base sequences are changed, it's called a mutation. A single mutation can have a large effect, but in many cases, evolutionary change is based on an accumulation of many mutations with small effects. Mutational effects can be beneficial, harmful, or neutral, depending on their context or location. Most non-neutral mutations are deleterious. In general, the more base pairs that are affected by a mutation, the larger the effect of the mutation, and the larger the mutation's probability of being deleterious. To better understand the impacts of mutations, we've considered many examples on beneficial and unfavorable effects of mutations that researchers have shown. Some mutations have a positive effect on organisms in which they occur. They are called beneficial mutations. They lead to new versions of proteins that help organisms adapt to changes in their environment. Beneficial mutations are essential for evolution to occur. They increase an organism's chances of surviving or reproducing, so they're likely to become more common over time. There are several well-known examples of beneficial mutations. To begin with malaria resistance, the classic example of evolutionary change in humans is the hemoglobin mutation named HBS that makes red blood cells take on a curved sickle-like shape. With one copy, it confers resistance to malaria, but with 2 copies, it causes the illness of sickle cell anemia. This is not about that mutation. As reported in 2001, Italian researchers studying the population of the African country of Burkina Faso found a protective effect associated with a different variant of hemoglobin, named HBC. People with just 1 copy of this gene are 29% less likely to get malaria, while people with 2 copies enjoy a 93% reduction in risk, as the gene variant causes, at worst, to mild anemia, nowhere near as debilitating as sickle cell disease. The second example is nylonase, nylon bacteria. Nylonase is an example of beneficial mutation in bacteria. The nylonase bacteria can eat short molecules of nylon 6. The mutation in these bacteria involves insertion of a single nucleotide in the genetic material. It's estimated that the frame shift mutation might have occurred in the 1940s, when nylon was invented. Nylonase can be used in wastewater treatment plants. The third is gene mutation in almond trees. Almond seeds from wild species contain amygdalin, a biochemical that converts into cyanide inside the human body. According to researchers, consuming wild almonds is fatal. A single gene mutation in wild almond trees resulted in variety that no longer synthesize amygdalin. When humans discovered this non-bitter almond species, they cultivated them, which is continued till today. Now to end with CCR5 delta 32 HIV immunity in humans. Cysteine chemokine receptor 5 is a receptor molecule located in the membranes of white blood cells and nerve cells. In a cell, CCR5 permits the entry of chemokines that signals the inflammatory response to any foreign particles. The gene responsible for coding CCR5 is present in a human chromosome 3. A mutation in this gene called CCR5 delta 32, involving deletion of 32 base pairs, affects the normal functioning of CCR5. In the initial stages of HIV infection, the virus normally enters through CCR5. However, mutated CCR5 blocks the entry of HIV. People carrying homozygous mutated CCR5 delta 32 are resistant to HIV, while heterogeneous ones are beneficial as they slow down the disease progression. Thus, CCR5 delta 32 provides partial or complete immunity to HIV. Imagine making a random change in a complicated machine such as a car engine. The chance that the random change will improve the functioning of the car is very small. The change is far more likely to result in a car that doesn't run well or perhaps doesn't run at all. By the same token, any random change in the gene's DNA is likely to result in a protein that doesn't function normally or may not function at all. Such mutations are likely to be harmful. Harmful mutations may cause genetic disorders or cancers. A genetic disorder is a disease caused by mutation in one or few genes. A human example is cystic fibrosis. A mutation in a single gene causes the body to produce thick, sticky mucus that clogs the lungs or blocks ducts in digestive organs. When a mutation alters a protein that plays a critical role in the body, it can disrupt normal development or cause a medical condition. A condition caused by mutations in one or more genes is called a genetic disorder. In some cases, gene mutations are so severe that they prevent the embryo from surviving until birth. These changes occur in genes that are essential for development and often disrupt the development of an embryo in its earliest stages. Because these mutations have very serious effects, they are incompatible with life. In addition to cancer, cancer is a disease in which cells grow out of control and form abnormal masses of cells. It's generally caused by mutations in genes that regulate the cell cycle. Because of the mutations, cells with damaged DNA are allowed to divide without limits. Cancer genes can be inherited. A single mutation will likely not cause cancer. Usually, cancer occurs from multiple mutations over a lifetime. That's why cancer occurs more often in older people. They've had more opportunities on mutations to build up. Many of the genes that contribute to cancer development fall into broad categories. Tumor suppressor genes. These are protective genes. Normally, they limit cell growth by monitoring how quickly cells divide into new cells, repairing mismatched DNA, and controlling when a cell dies. When a tumor suppressor gene mutates, cells grow uncontrollably, and they may eventually form a tumor. Klinefelter's calicose. Klinefelter syndrome, also known as XXY syndrome, is a genetic mutation in which a male subject carries an extra X chromosome, therefore carrying the male genotype XX in addition to the traditional male genotype XY. Likewise, males with the Klinefelter syndrome often have feminine features, such as breast tissue, and may not be able to reproduce. As it lies in the genetic code, which is homologous between most species, Klinefelter syndrome is not exclusive to humans. Therefore, cats, dogs, and even whales can inherit the XXY genotype. In cats, the X chromosome carries more than sex-related information. Fur color, for instance, is carried on the X chromosome. Furthermore, fur color is codominant, as a male cat typically inherits only one X chromosome and female cats inherit two X chromosomes. Female cats are more likely to have multicolored fur patterns than male cats. This is especially true for the calico, a cat known for its striking orange and black fur. The gene for black fur can be carried on the same X chromosome as the gene of the orange fur, which makes calico cats almost exclusively female. However, this doesn't make the existence of a male calico impossible. Male cats with two X chromosome or genotype XXY may very well carry the gene for orange fur on the X chromosome, and the gene for black fur on the other one. In this way, they are indeed Klinefelter's calicos. Although the CCR5 delta 32 mutation has one drawback, it is strongly associated with a chronic liver disease called primary sclerosing cholangitis, a long-term progressive liver and gallbladder disorder characterized by inflammation and fibrosis of the intrahepatic and extrahepatic bile ducts, causing the bile to drain from the gallbladder.